So I wanted to talk to you today about what we would have done for our short tandem repeat experiment in class. So this is a general lecture on how the police department uses DNA to establish identity. So, just as a reminder, long ago they noticed that certain genes were linked and tended to be inherited as a group, and that's originally how we learned about chromosomes. And just as a reminder, if you've forgotten from your basic biology, humans have 23 pairs of chromosomes. Now, within our chromosomes, a large amount of our DNA is repeated sequences that do not encode genes. We have clusters of genes, and then we also have large areas of our DNA that would have GATC, 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 certain numbers of repeats, but don't actually code for anything. And those are free to mutate. And they're actually, because of slippage in the number of repeats, they can from over time and potential mutations from father to child, you can get some errors in the DNA replication creeping in. So over multiple generations, you can have, one particular site that a father might have 8 repeats and the son might have 9 repeats or something like that. But that's still fairly rare, and it maybe would take, one mutation every, 30 generations or something like that to be seen and to start to become commonplace. So for the most part, all of your number of repeats, your repeating sequences in your DNA are inherited from your parents. So this is how paternity testing works, and this is how genetic identity. There are islands of genes, and it's the repeating sequences in our DNA that forensic science is using to be able to genetically identify an individual as matching a sample left at a crime scene, for example. Only 0.1% of DNA actually differs from one person to the next, but within that 0.1%, that's all of our, whether you have curly hair or straight hair, blue eyes or brown eyes, do you have a wide nose, do you have a cleft in your chin, all of those differences all come from that 0.1% of DNA. Also in that 0.1% of DNA is the number of repeats that you have in specific areas of your DNA. Now, there are thousands and thousands of these different locations in your DNA where you have repeating segments of your DNA, and not all of them are useful. So labs around the world over time have looked at all these repeating segments of the DNA, and they've said, okay, the ones that are useful are the ones that have a large amount of variability between people. So one of the downsides of having blood types, mostly everybody is type O or type A. Very few people are type B or AB. So if you find type O blood at a crime scene, it's pretty useless because 43% of the people have type O blood and 42% of the people have type A blood. So, you find type O or type A, you're like, well, most of the people are still included in this type of blood. So scientists started looking at the DNA to be able to generate a DNA profile of an individual, and they looked at these variable regions in the DNA to do that. So of these 10,000 regions that are variable in terms of the number of copies, they chose a few, and some of their criteria were you had to have varying numbers of these repeats, the tandem repeats one after the other, GATC, GATC, GAT, etc. But also, outside of these repeating areas, there had to be constant regions of the DNA for every single person. And that's important because they want to amplify these by doing PCR, which means you need to be able to bind a PCR primer to it. So you need to be able to have a kit that contains a PCR primer that is going to bind to everybody. You can't be making individual PCR primers for all these different people. You have to have one set that all police departments across the entire U.S. can use. So they use the variable regions to generate a DNA profile, and you might leave lots of different types of DNA at the crime scene. Maybe you leave blood at the crime scene, or maybe if you're an archaeologist, you have an old piece of bone and you want to be able to generate a DNA profile of an individual from bone. Maybe you find hair which has a root on it and has been pulled out of somebody's head. As long as that hair is not naturally shed, it pulls out the follicle. You have some genomic material from that hair that you can then put through this short tandem repeat process and generate a DNA profile of an individual. So many other body tissues or body products have DNA in them. Some of the body products, semen. So if you have a rape case, you've left your DNA behind. Saliva, somebody spits or licks something, you have DNA from your cells in your mouth. Urine, very often, more often than you would think, burglars are nervous before they rob somebody's house. So they will take a pee against the side of the wall of somebody's house. And after the burglary happens, the police department will look for these unusual spots, and they'll try to isolate. They'll try to do swabs to see if they can get some DNA from something that the criminal has left behind. So as I said, hair, teeth are an excellent source of DNA. A nice sterile contained environment in a tooth, bones, tissue, lots of different sources of biological evidence. Now, when you have cells that you find at a crime scene, there are two different types of DNA you have in each cell. You have your nuclear DNA inside of the cell, and you have two copies, two of each chromosome inside the cell, and you inherit one copy from each of your parents, one from your mother and one from your father, and that's unique to a particular individual. You also have mitochondrial DNA, and you have many, many copies of mitochondrial DNA in the cytoplasm area of your cell, and these are all identical and all inherited from your mother, and they're not unique to an individual, because if you have brothers and sisters, they're going to have the exact same pattern of mitochondrial DNA that you do, assuming you have the same mother. Now, within these two types of DNA, there's different types of polymorphisms. Within mitochondrial DNA, you have what's called a sequence polymorphism. And so a sequence polymorphism means there's been a particular base change right here. And this base change is something which can indicate, where you came from a long time ago, because certain groups that were consisting of people who married within tribes tended to have particular, very similar mitochondrial DNA patterns in them. This is also the kind of thing that 23andMe looks for, both in your mitochondrial DNA, and you also have some sequence polymorphisms in your genomic DNA that they look for, particular bases that are associated with eye color or, again, genetic heritage. So sequence polymorphisms are what's used by archaeologists and used by genetic testing companies, but the police department is only interested in the size polymorphisms. So these size polymorphisms is basically, a particular sequence that is repeated multiple times. So consecutive, repeated, identical sequences, and they're in tandem, they're right next to each other, and they're very short. So they're called short tandem repeats, or STRs. So remember that I said that they particularly looked for varying numbers of repeats between people. while having areas that the primer is going to bind to be constant among everyone. So basically, you can bind primers to these constant regions of the DNA. And when you bind primers and you amplify between these primers, the only thing that's going to be different is how many repeats somebody has. So if you have 7 repeats, this is going to be a particular size. But if you have 8 repeats in here, it's going to be a little longer. It's going to be basically, if you have 1 extra AATG, it's going to be 4 bases longer. So when you're looking at short tandem repeats, the repeat region is variable between the samples, but the flanking regions where the PCR primers are binding is what it has to be constant so that they can have one kit that everybody can bind to and everybody can do the exact same PCR reaction. And all they then need to determine is what is the overall size. So when the police department is establishing genetic identity, the majority of the tests that they do, and we'll talk about the minority later on, but the majority of tests they do, they're only interested in what size is this fragment. Now, you can be either a heterozygote or a homozygote. And in a heterozygote, the alleles are differing in size. So remember, you get 1 chromosome from your mother and 1 chromosome from your father. So you can have gotten a 7 from your mother and an 8 from your father, or an 8 from your mother and a 7 from your father. So there's 2 different ways you could be a 7 and an 8. But if you're a homozygote, it means that you'd be a 7 and a 7. You would have gotten the same number of repeats from both of your parents. So you can be either a homozygote or a heterozygote at the particular site of DNA that they are looking at. So these are the original 13 CODIS, or Combined DNA Index System, that the FBI used to genetically test people. And they have names like CSF1PO and THO1 and VWA, but eventually all the DNA scientists got tired of trying to remember, where exactly is the VWA versus the THO1 versus the CSF1PO. And they said, we need to come up with a better nomenclature system. So they all got together and said, okay, D7 is DNA from chromosome number 7 at location S820, for example. Or this set of markers is chromosome number 8, location S117 on chromosome number 8. So basically, they got together and they standardized the nomenclature, but some of the original nomenclatures still exist just because they were already published under those particular names. Now, there's also the amylogenin gene, which actually, interestingly, codes for tooth pulp and is different sizes between males and females. So they can also do a PCR on this and say, well, do we have, only one side? Is it a homozygote, in which case it's an XX and a female, or is it a heterozygote with 2 different sizes, in which case it is an XY and it's a male? So overall, after they do that PCR reaction on it, they're going to run it on a gel and separate it out by size. So they'll be able to tell what the size of it is. And they have a set of DNA markers. Now, it takes a fair bit of effort and a little bit of time. It takes on the hours time range to do a PCR reaction and separate the various sizes in the gels. So rather than have the police department sit there and do 13 different PCR reactions, companies have developed the ability of using different primer sets in the exact same reaction that are producing different PCR product sizes, so they can do 2, 3, 4, etc. Different STRs. They can look at all these different STR sites all at the same time on one gel. So you can have on chromosome number 7, for example, you can have a 7 repeat and an 8 repeat here. On chromosome number 8, you also might be a 7 repeat and an 8 repeat. So how are you going to tell the difference between chromosome 7 with 7 and 8 repeats and chromosome 8, maybe with also a 7 and an 8 repeat? Well, the answer is by putting the PCR primers in different locations. So if the PCR primers are very, very close at chromosome 7, you're going to have an overall smaller size. If you put them farther away at this PCR location that you are amplifying, you're going to have overall larger sizes. But you can still tell the difference between these because they're going to be in completely different areas of the gel. And so you can actually do what's called a multiplex PCR, where you can be copying multiple markers at the same time. And actually, you can do up to 24 markers at once now. Used to only be able to do 16, but now you can actually do 24 different markers. I showed you the original 13 CODIS loci used by the police department, but as of this past year, the police department increased their number, and now they're checking 24 different sites on the DNA so they can get much higher statistics in court. It's sensitive. It can be used with levels of less than 1 nanogram of DNA. So basically, if you touch something with a fingerprint, you're going to leave behind maybe 10 cells, maybe more. That touching of something with your fingerprint that leaves behind 10 cells, that's enough to get a genetic profile using this methodology. And how they do this is they use different fluorescent dyes to distinguish all these alleles with the overlapping size ranges. And I'm going to show you what that means in just a minute. So this is a picture of a gel. So this would be 6 different people. So this is person number 1, person number 2, person number 3, person number 4, person number 5, and person number 6. So 6 different individuals. And in this case, 6 different individuals have been tested at 8 different sites on their DNA. And these are the size range markers right here. So for CSF1PO, that's one of those size range markers that I talked about earlier in the CODIS loci. So at CSF1PO, the majority of people have between 6 and 15 repeats in their DNA at that location. Compared with, let's say, VWA, the majority of people have between 11 and 21 repeats in their DNA. But notice you can have 11 and 21 repeats down here, and these are all in a very different size range than up here. You can have some overlapping numbers of repeats in the TPOX locus. So most people vary between 6 and 13 repeats at the TPOX locus, which you'd say, well, how am I going to be able to distinguish between 6 and 13 and 6 and 15? The answer is, where are all of these primers found relative to these number of repeats? So at the VWA locus, the primers are very, very close into the repeats, so all the sizes are generally smaller. In CSF1PO locus, the primers are farther away from the individual numbers of repeats, so the sizes are generally much larger. So they're all separated in a gel, but they're all in their own individual little size ranges. And it's also important to realize on this gel, they're loading these size range markers. So if something, so for VWA, for person number 6, for example, you have one band right here and one band right here. We're not looking about that and that. That's from person number 5. This is person number 6 right here. So the person number 6, we go down to VWA site, and they have right here, and they have right here. So here we have 11 repeats, 12, 13, 14, 15, 16, 17, 18. So we would put somebody into kind of like an Excel spreadsheet. We'd say, okay, at VWA, person number 6 is 16, 18. And if you then go to THO1 for person number 6, at THO1, there are 2 bands here. And that's going to correspond to 5, 6, 7, 8 right here. 9, 10, 11 if they're higher. So this person would have a 9 right here. So between 5 and 11 repeats. So person number 6 for THO1 would be put into the system as a 6, 8. And this is heterozygous. On the other hand, at TPOX, person number 6 would be put into the system as an 8, 8. So they only have a single heterozygous location, and they only have 8 repeats. So this means they inherited 8 repeats from their mother and 8 repeats from their father. Whereas here, they could have inherited a 6 from their mother and an 8 from their father, or an 8 from their mother and a 6 from their father. 2 different ways that could have occurred. And so you can go through and you can then categorize them at the location of CSF1PO. And that's nice. You can do maybe 4 on the same gel, but if you tried to do more up here, they'd get so close together you wouldn't even be able to tell the difference between all these sizes. So what do you do if you want to do more than 4 size ranges on a gel? What you do is you use different fluorescently tagged markers. So these are all tagged in red, for example, and all of these are tagged in green. So you can see the reds and the greens are overlapping on the gel, but they can all be run in the gel at the same time because they're overlapping to our eyes, but not to a computer. You can say, computer, show me only the red. Computer, show me only the green. And so by having only 2 different fluorescently tagged markers, then you're able to have 8 all running in the same, from the same tube on the same gel. Likewise, if you were to use blue fluorescently tagged and yellow fluorescently tagged and a number of different colors with different fluorescent tags, that's how, that's how they're able to do 24 different locations on the DNA all at the same time and all in a single tube. So once we have the statistics, once we know that VWA is a 1618 and THO1 is a 68 and TPOX is an 88, how do police use those numbers to be able to say, well, what's the genetic identity? What's the probability of this person matching this DNA that we found at a crime scene? Because you can't just go into court and say, well, it's a match. You have to be able to explain to them how you know it's a match and what's the probability against anyone else having the same pattern. So how they do that is just using the probabilities. So there are data tables that were compiled by genetic identity companies like Promega that determine the probability of having each of these numbers of repeats in these short tandem repeats. And there are tables for different major race designations. So you can have some probabilities if you're Caucasian American, African American, Hispanic American, Asian American, etc. Some other groups they're working on, but they have pretty good tables for the major race designations. And as you can imagine, someone who's African American, which means they have significant Caucasian ancestry, is going to have slightly different probabilities than someone who's of straight Nigerian ancestry living in Africa. So all these types of things are taken into account when they are using these tables of probability. So here's an example. Let's just say it's Caucasian American probabilities. And we already said that person number 6 at VWA was a 1618. So the probability of being a 16 is 0.211. 21% of the people have at least one 16 in their genome, 16 repeats. And 20.2% of the people have at least one 18 in their genome. But the probability of being both a 16 and an 18 is statistics. So 2 times the probability of the first times the probability of the second. The number 2 is important because as long as you have differences in there, there's 2 different ways you could have inherited. You could have gotten a 16 from your mother and an 18 from your father, or vice versa. So 2 different ways that you could be a 16-18. So 2 times the probability of the first times the probability of the second is 0.08524, or 8.5% of the people would show up as having both a 1618 at the VWA locus. So now let's do THO1. So we'd said that the THO1 person was a 6-8. And so we look at the data table for THO1, and the probability of a 6 is 0.237. The probability of an 8 is 0.117. So 2 times the first times the second is 0.055, or 5.5% of the people are going to potentially be a 6-8. But what if you're both a VWA 1618 and a THO168? So again, you're multiplying those two probabilities together. So even if we've only looked at a person at two different locations on their DNA, we've already gotten down to, when we multiply those two together, we've gotten down to 0.0047, or 0.47% of the people would be both a VWA 1618 and a THO168. So we've only checked two locations, and already we're down to less than 0.5%. So if the FBI repeats it at 13, 16, or 24 different locations, they're able to get numbers like the probability after they finish checking 24 different locations on the DNA is 1 in 500 trillion that anybody else would have the same genetic pattern throughout their entire 24 locations that we're checking. So this pattern in crime scenes is then submitted to CODIS, the Combined DNA Index System, and there's different levels. You can submit it to the local, state, or national level databases, and it contains profiles from offenders, profiles from crime scene evidence and missing persons, so that if you have a match, you can use that match to link crimes to each other. So if all you have is crime scene evidence, but you find the same pattern at a crime in Arizona and a crime in Florida, that says, okay, now we're looking with someone who's traveling between Arizona and Florida. So they use this to catch one killer one time who was going up and down I-5. There was similar evidence found in Washington, in Oregon, and in Northern California, and on some bodies. And so by knowing that this person traveled up and down I-5, they started looking at long-haul truckers. And so a long-haul trucker was eventually found and convicted of killing prostitutes because he left his DNA behind, and they were able to link the crime scenes based on the genetic material that was left behind. So the match can link crimes to each other, or if you have a suspect, they can be linked to the offender. Now, an easy way to link them to the offender is many offenders are in the system. So everyone who's currently arrested for a felony has to submit their DNA into the system. If you are found innocent of that felony, not guilty, you are allowed to petition to have your DNA removed from the system, but a lot of people don't bother to do that because they don't know it's available to them. But if they're found guilty, that's staying in the system no matter what. Now, there's other types of DNA analysis that are done with other different types of the polymorphisms. You can have short tandem repeats on the Y chromosome, and those are good for tracing patrilineal heritage, and this is something that's done by 23andMe. You can choose to trace your maternal ancestry. You can choose to trace your paternal ancestry, and if you're choosing maternal ancestry, 23andMe will use mitochondrial polymorphisms, and if you're trying to trace your paternal history, they will use the short tandem repeats found on the Y chromosome. Now, there's also single nucleotide polymorphisms, and as I said before, these are those single base changes that appear. Mitochondrial DNA was one example, but you also have that in you also have that in your genomic DNA. Now remember, when you are talking about mitochondrial DNA, it is maternally inherited. So if you have a female, any children of that female are going to have the same mitochondrial DNA. It's passed down through the maternal line. So any offspring of these same, so from the grandparent to the daughters, for example, all of these cousins are going to have the same maternal inheritance. But if there's a son, while the son is going to share the same mitochondrial DNA, the children of the son are going to have the same maternal mitochondrial DNA as their mother, and so the father's line dies out. So this is just a reminder of how the patterns of inheritance go. So this is one of the things they're looking at in mitochondrial DNA. There's hypervariable regions in the mitochondrial DNA, and because they don't actually code for any particular genes, they're able to mutate somewhat more frequently, and so mutations over time have crept into them, and so they're actually sequencing the mitochondria. So if this is a normal sequence, they would compare the sequence that's found to a normal sequence, and they'd say, okay, we have a mutation at position number 263, and in the original sequence it was a T, but here we have a mutation, and so we're going to call it 263C. And so you can be put into a database like that, and then this is how large-scale migration patterns have been determined by anthropologists from looking at the DNA of a number of different people, because the mitochondrial DNA mutates at very predictable rates, about one mutation every 30 generations. The more mutations you have in your DNA when compared to some early DNA patterns, so the more mutations you have, the longer time it's been since you split off from that particular group. And then they're able to say, well, how many migrations there's been, because if there's one migration, these people who migrated earlier are going to have some mutation that the people who migrated later are not going to share. So this is how anthropologists are able to determine, that there was an out of Africa event that occurred a certain number of years ago into Europe, and then there was another one where people traveled south along the continents of India and managed to make their way to Australia. Further events pushed people into the China and Russia region, where they crossed the land bridge at several different times into the Americas in several different migration events. Now, single nucleotide polymorphisms are also used as well as being used in genealogy, they're also used in things like personalized medicine. So if you have breast cancer, for example, one of the things the doctor's office will do is they will do that. They will sequence the type of cancer you have, and they will see, okay, what single nucleotide polymorphisms do you have in this cancer? And that tells them what kind of medicine your cancer is going to be more responsive to. So if you've ever listened to those commercials that say, if you're this type positive and this type negative, here's a drug for you. So this is what the doctor's offices are using. They're saying, do you have this particular mutation? Do you not have this particular mutation? And then using that to prescribe the best type of chemotherapy that has the best chance of working against your particular type of cancer. So another thing that police departments are using these single nucleotide polymorphisms for, they're using these genealogy websites to now search family groups. So they would have DNA that is at a crime, and they have had no luck putting it into their regular databases. But what they did in the case of the Golden State Killer, as well as some others, they anonymously submitted a sequence of DNA to genealogy websites, and they didn't find the exact same person, but what they did is they found people who were closely related to someone whose DNA was left behind at a crime scene. So then they use the genealogy websites the same way that people use 23andMe to find, parents, cousins, things like that. They did this searching for family members, and through genealogy they were able to say, okay, well, chances are it's maybe one of these three men that are cousins, because they knew it was male DNA, because they could sequence the amylogenin gene and say, okay, two different sizes, so there's an X and a Y chromosome here. And so then they narrowed it down to cousins. They knocked on the door of a guy and said, hey, can we have a DNA sample? He went, sure, you can have a DNA sample. And so it didn't match when they compared DNA samples. And so they went on to the next guy. And then they got to a guy they were pretty sure who it was, but they didn't want to alert him that, because he was a former police officer, so he knew how things were done. So they didn't want to alert him that he was under suspicion. So what they did is they followed him and they anonymously got a DNA sample from him. Anytime anything leaves your household, anytime anything leaves your body, it's fair game. So if you put trash out by the curb, the police department is allowed to search your trash. If you spit on the sidewalk and they see you spit, they can go up to that sidewalk after you've walked away and they can collect that phlegm or that saliva from the sidewalk and they can search that. And it's enough when they get a match. It's not going to hold up in court, but it's enough for them to say, hey, we saw this guy spit on the sidewalk. We collected the sample from the sidewalk, and it matches this crime scene sample. So, judge, we'd like to get an arrest warrant for this particular person. And then once you have the arrest warrant for the person, then you can force them to submit their DNA in the database. And then you make the official determination using the short tandem repeats and get the 100% match that will stand up in court, that you even have statistics for. Another thing that they're doing with DNA, when they have a completely unknown person, what they will do is they will submit the DNA to one of these companies that is working on what's called phenotyping. So based on your DNA, they know that particular single nucleotide polymorphisms or particular sequences in your DNA are associated with various body types. So the first thing they'll do is they'll say, okay, well, what's the ancestry of this person? And, once they're pretty European, they'll say, okay, the ancestry is European. But then they go one step further and they say, okay, particular sequences in your DNA are associated with skin color. Particular sequences, particular SNPs are associated with the probability of having particular colored eyes. So they can say, well, not brown eyes, not blue eyes, but maybe somewhere in the middle. And they can say, not red hair or blonde hair, but probably a light brown hair. How many freckles do you have? So they're able to say, do you have a cleft chin? Do you have earlobes that are connected or that are continuous down into your side of your face? Do you have a wide nose? Do you have a narrow nose? So they're working on being able to make a picture of the person to be able to help the police department. So when you discard a sample of DNA at a crime scene, if the crime is heinous enough, the police department will send a sample out so that they can say, what is this? What are we looking for in this person? So hope you've enjoyed this lecture on short tandem repeat DNA.